So we left here yesterday at the vitamin K. We have studied it completely. And today we're going to start with the zinc, the function of zinc. You should know that zinc is a very important cofactor for, many enzymes like metalloproteinases. And you should know that metalloproteinases are the enzymes which are required for the proper wound healing. You will study it later into the packed section when you study about this wound healing part, and you will see that metalloproteinases requires the zinc as a cofactor for their function. Plus, many intercellular receptors are there inside your cell, which also requires this zinc from the motifs. They carry some intercellular receptors in them, so that's why zinc is a very important thing, inside your body. What would happen if there is a zinc deficiency occur inside your body? So one of the syndrome is acrodermatitis enteropathica, which is related to the defective intestinal absorption of your zinc. And the patient will get this dermatitis, which is your skin rash, enteropathica, जिसके अंदर आपका आता है diarrhea, and you will also get this hair loss and the loss of taste sensations. And you should know the guys that these skin rash are generally around the body orifice, for like your around your mouth, you know, around your anal canal and other areas, the body orifice. So you will also get this delayed wound healing because I told you that zinc is a cofactor for the metalloproteinases which are required for the wound healing. So you will get a delayed wound healing. You will get very low immunity because of this receptor thing. You will get the male hypogonadism. You will have this loss of hairs, facial hairs, the axillary hairs, the pubic hairs. You will get this dyscrasia, which is a loss of taste sensations. You will get the anosmia, which is loss of smell. And due to this anosmia, you will also get this loss of sensations, the taste sensation also. Okay. And you should know, guys, this zinc deficiency can also predispose the patient to the alcoholic cirrhosis. Any doubt? You should remember this thing, the acrodermatitis enteropathica, which is due to the defective absorption of zinc in the GIT. And this leads to diarrhea, skin rash, hair loss, and loss of smell, loss of taste sensations. Patient might even get this hypogonadism thing. So you will get, the infertility. Okay. And you will get the delayed wound healing. Now we will move forward for the protein energy malnutrition. And you should know about these two syndromes, the Kawashioka syndrome and the marasmus syndrome. Marasmus is kind of, less severe than the Kawashioka. Okay. Because in this thing, your protein malabsorption is occurring, and as a result, all of your oncotic pressure inside of blood is losing. You require the proteins inside your blood to maintain the osmotic pressure, right? And if you lose the proteins, then you won't get this, the oncotic pressure, and so you will get the loss of blood volume, and your blood will start accumulating inside your, extracellular spaces, and you will get this edema thing. You will get the swollen abdomen. Okay. You will get this fatty change because of the fact that you require these apolipoproteins to function, right? And if you lose this apolipoprotein, then what will happen? Your fat will start accumulating inside your liver. You will learn about this apolipoprotein thing in the later chapter in this metabolism. Okay, so what you will get is a liver malfunction because of loss of apolipoproteins. Due to the loss of proteins, you will get this loss of oncotic pressure, and you will get the edema. Okay, so yeah, that's about the Kawashioka syndrome. And then the marasmus, which is kind of less severe than the Kawashioka. You have some protein inside your body, and that's why you do not get this edema thing. Okay, so you should remember the muscle wasting is occur in the marasmus and edema is occur in the Kawashioka.
Can I ask?
Yeah, please.
Why does the skin lesions occur in Kwashiorkor?
It's just a symptom, really, because as I told you, right, the zinc is involved in hundreds of enzymes, right?
No.
And
I'm asking about Kwashiorkor.
oh, where?
The
It is
protein malnutrition.
it is completely malnutrition thing. Your body is not having any energy to to do any function. You do not having any protein. Your collagen. Remember, you require the collagen. You require the protein to function, right? And it is very important component of your skin. The collagen, you will lose it, right? So all of these things are contributing to the skin lesions. It's not just a single thing, but due to many factors combined.
All proteins are
But
deficient.
yeah, complete deficiency of protein is happening in the Kawashioka, complete protein deficiency. And that's why you're even losing your oncotic pressure. See, as you can see, this child, he's having this edema of the abdomen, as you can see here. And this is your marasmus, which is a muscle wasting you're happening. Okay, you just need to remember this fact that the edema will be in the Kawashioka and this non-edema is your in the marasmus. Okay, this is the important thing.
Also,
Yeah, someone saying something? Any doubt? Should I move forward now?
Yes, you can. Somebody has not muted its mic, so it's a background noise.
Okay, let's study about this ethanol metabolism, guys. Remember, I told you one thing in the peroxisomes part that it is involved in the ethanol metabolism. So you see here this thing, the catalase enzyme, which is involved in the ethanol metabolism. As you can see, the peroxisomes are also involved in the ethanol metabolism, and similarly, the microsomes are involved in the ethanol metabolism. The first thing you should remember that in the ethanol metabolism, you are using all of your NADP and NAD stores. All of this NADP is being utilized to the NADP, and all this NAD is being.
ఈమె చేయించుకుంది కదా చూడు నాకు ఇలాంటి డాక్టర్ అవ్వాలని డబ్బులు సంపాదించవచ్చు
Who is this?
ఈమె వీళ్ళ వైఫ్ కానీ నేను అది ఈమె ఎవరి మాట్లే ఇక్కడ ఈమె జాలెన్స్ ఎలా ఉందో
Please mute your mic.
ఇక జాలెన్స్ ఎలా ఉందో ఈ పిక్చర్ ఎలా ఉందో
This is Nayan member.
It's Nayan, right?
Yeah, you can mute its mic.
ఎలా ఉందో
You can.
Nayan?
అవును.
Okay, let me mute her. Okay, I've muted her. Okay, so the ethanol metabolism part. You should remember this one thing that all of the body NADPH store is being utilized to the NADP. Okay, converting into the NADP, and all the body NAD store is converting into the NADH. So you see all these symptoms that's going to happen in your body after the ethanol consumption is due to the fact that all the NAD stores is being utilized completely to the form the NADH. So every reaction gonna reverse, every reaction gonna stop, and all the reaction are gonna divert to one thing, is to form this NAD stores back. Okay, and because you're using this NADPH from your body, your body is no longer protective from this, this reactive oxygen species, right? So you're gonna get damage from this reactive oxygen species. Okay, so you should know this reaction, the ethanol with the help of enzyme ethanol dehydrogenase will be
the last point, please.
Yeah, because you're losing your NADPH store completely from your body. Ultimately, what's going to happen, this NADPH is required to fight the reactive oxygen species, right? And because now you're depleting your NADPH store, what's going to happen now? You won't get fight from this reactive oxygen species, right? So this damage will occur due to this reactive oxygen species because of this loss of NADPH from your body. Okay, this thing you should remember about this NADPH thing. And the NAD is very important because you're losing complete store of NAD from your body. So all these reactions are going to divert to one single thing, which is to form the NAD again. Okay, now this ethanol is converted to the acetaldehyde with the help of this enzyme alcohol dehydrogenase. And you should know that this acetaldehyde is very toxic for our body. All the symptoms, all the acute symptoms that you will get after the ethanol consumption is due to this acetaldehyde, the abdominal pain, the vomiting, the flushing reactions. All of these things are due to this acetaldehyde. So as soon as this acetaldehyde form, it need to be converted into the less toxic thing, which is your acetate, which is the acetic acid. Okay, and it is done by the enzyme acetaldehyde dehydrogenase. Okay, as you can see, this enzyme requires NAD to form the NADH, and similarly, this enzyme requires a NAD to form the NADH. And you should know about this drug, the disulfiram, which you give, as to control the ethanol, addiction, right? You give the patient disulfiram. Okay, what would happen? You're actually going to inhibit your acetaldehyde thing, right? So ultimately, what will happen is this acetaldehyde, which start accumulating inside your body, and you will get all these symptoms like abdominal pain, the facial flushing, the nausea, the vomiting thing. So this disulfiram for alcohol addiction. Okay. Second thing you should know about this fomepizole. Once you get any, toxicity from this ethanol or the methanol or the, ethylene glycol. So what will happen? They will get converted into this toxic metabolite, right? So you will give the patient fomepizole to stop this conversion. Okay, this is a very important drug, fomepizole, for this overdose of methanol and the ethylene glycol, and disulfiram to discourage the drinking. Because it increases the hangover symptom due to this acetaldehyde thing. Okay. Now you should know about these reactions. What's gonna happen when you consume, too much of alcohol for a long duration. So as I told you that your NAD stores are gonna deplete very soon.
There isn't
So
going to come
ultimately,
any question from microsome?
microsome, no. I never encountered any question on the microsome, but I do encounter a question on peroxisome about this ethanol. So you should remember that these organelles are also involved in the ethanol metabolism.
Like
And they
they're
have
not
this enzyme
dependent.
known as like, yeah.
Like we would not be supposed to remember the exact number cytochrome P2E1 for this enzyme metabolism.
You will study about these enzymes later, actually, in the pharmacology section, general pharma section. I will tell you about these enzymes, all their numbers, and what substrate they use, what substrate they convert. Okay.
And
So
we
you
are
will
supposed to
remember.
remember the numbers also.
Yeah, exactly. You will supposed to remember the numbers. But there's a trick, a slight trick I will tell you to remember all these things. Okay, don't worry about that now.
Okay.
Just know that microsome is involved, the peroxisomes are involved, and these have this enzyme as a catalase which are involved. Okay. So now let's talk about all these reactions which are going to take place once you consume too much of alcohol. So I told you that on all these NAD stores are going to deplete. And so that all of the reactions are going to divert to one thing, is to form this NAD back. Okay, so you already know about this glycolysis thing, all the glycolysis were gonna, stop. Okay, because it uses the NAD to form the NADH. So it will stop. What happens is now you see about this, you will study about this cycle later in the metabolism part. You will see that this malate converts to the oxaloacetate and will requires NAD to form the NADH. So this step is going to inhibit. And what will happen? The opposite thing. The oxaloacetate going to convert it into the malate, and all this, this it will use NADH to form the NAD again. So that's how the one thing you will get the NAD. Secondly, your pyruvate will going to convert into the lactate so that it could regenerate this NAD back. And that's why in the alcoholism, you will get this lactic acidosis, which is a metabolic acidosis, and anion gap metabolic acidosis, because you're accumulating extra acid inside your body. Okay. Your DHAP will convert it into the glycerol 3-phosphate, which is utilized for the formation of triglycerides. And so that again, you will regenerate your NAD stores from here. So as a result, as you can see, because all of this acetyl coenzyme are going to convert it into the fatty acid and the keto acids to form this NAD, you will get very high fats in your body. Okay. And that is the reason you will get this hepatic steatosis in the alcoholism. Okay. You will get too much of keto acids inside your body again in the alcoholism. Okay. So you should remember these are the pathways. The dotted pathways are going to stop because all of them requires the NAD to convert it into the NADH. So all these pathways are going to stop, and the pathways which form the NAD are going to initiate too much, are going to become super active. And as you can see, all this glucose is going to utilize to form the oxaloacetate and then the malate. You will get this fasting hypoglycemia because now you're not having any gluconeogenesis, right? Because it requires this step, the conversion of oxaloacetate into the phosphoryl pyruvate. So this step is not going to happen. You won't get any new glucose formation. So you will have this fasting hypoglycemia. Why I'm saying the fasting hypoglycemia? Why not the instant hypoglycemia? Anyone? Why the fasting hypoglycemia? No?
Gluconeogenesis happens in fasting.
Yeah?
Gluconeogenesis happens in the fasting state.
Yeah, it will happen later in the pathway, of course, but why not the instant hypoglycemia is happening? Why the fasting hypoglycemia only?
Is instant for
Glycolysis is not.
with insulin?
It's about this glycogen. It's about the glycogen because you have this. Yes, exactly. You have this glycogen stores inside your body, which will utilize, just after the fasting. But later on, when these stores completely depleted, you won't get any new glucose formation, and so that you will get only the fasting hypoglycemia, like uh, hours later, 10 to 20 hours later, you will started having this fasting hypoglycemia, not the instant one. Okay. So I told you about this lactic acidosis thing in the alcoholism because you're gonna need this NAD formation again in order to continue this glycolysis thing because you require the energy, right? So glycolysis must be happening. You will get this NAD and you will get this lactic acidosis, which would lead to the high NN-gap metabolic acidosis. You will get this fasting hypoglycemia because you stop the conversion of oxaloacetate into the phosphoryl pyruvate. All this oxaloacetate going to convert into the malate. So you will get this fasting hypoglycemia thing. You will get the ketoacidosis. Okay, because your TCA is stopped. So SL coenzyme will go and form the fatty acids and the keto acids. You will get the hepatosteatosis because there's an increased production of this glycerol 3 phosphate and there's an increased production of fatty acids. You will get the hepatic steatosis. Okay. And you should know that this NADH by NAD ratio is going to go so much high so that all these pathways are, going to focus only on one thing is to regenerate this NAD stores. I hope you guys understand this thing about the alcoholism. Okay. And that's why all these symptoms of alcoholism, going to happen inside your body. Any doubt? No? Moving forward now.
Yes.
And that is yeah.
Well, you just said that fasting hypoglycemia is named so because first of all the glycogen stores will be depleted in an alcoholic,
Firstly, yeah.
and then
Yeah.
and then there will be no further glucose available. So all the pathway will divert to conversion of NADH to NAD to restore that rather than making glucose.
Yeah, exactly. Because you
So
want your glycolysis to be continued, right? You need the energy to function. And this is especially about the RBCs because RBCs do not have this step or this step, they're dependent only on this step. Remember the glycolysis thing? RBCs are dependent only on this thing. So you record especially these steps because of the RBCs.
So, okay. And
Okay.
there is no gluconeogenesis while the person
No,
is
no. As you can see, this conversion is stopped. As you can see, oxaloacetate is not converting into the phosphoryl pyruvate because you require this NAD. So you get an increased malate in your body, you will get increased lactate in your body, you will get increased fatty acids, keto acids in your body. Okay.
Okay.
So now moving forward for the biochemistry metabolism, you should know some sides of some reactions. First of all, the mitochondria. You already know that TCA cycle takes place in the mitochondria. The electron transport system takes place in the mitochondria. The oxidative phosphorylation takes place in the mitochondria. Remember, and the beta oxidation of the fatty acids you will study is takes place in the mitochondria. The ketogenesis you will see later is takes place in the mitochondria, and one of the important enzyme, which is your CPS1, is present in the mitochondria. Remember the location of CPS1 and CPS2.
Cytoplasm.
Yeah, CPS2 in the cytoplasm, CPS1 in the mitochondria. You should remember the location. Okay. And you should know the carboxylation of pyruvate, which is this step, the conversion of pyruvate into the oxaloacetate. This conversion from pyruvate to oxaloacetate here. In the gluconeogenesis, you will see the pyruvate first convert into oxaloacetate, oxaloacetate convert into the malate, malate then again go out of the mitochondria, enter the cytoplasm, it will get converted into oxaloacetate, and finally this oxaloacetate converted into the phosphoryl pyruvate. So this reaction, the conversion of pyruvate into the oxaloacetate. Can anyone tell me what enzyme is there in conversion of pyruvate to oxaloacetate? I told you in the vitamin section about it. The enzyme that converts the pyruvate into the oxaloacetate.
Pyruvate carboxylase.
Pyruvate carboxylate. What vitamin it requires? The carboxylation reactions?
B1.
B1.
No, not the B1. It was dehydrogenase. The carboxylation reactions. Vitamin B7, remember?
Biotin. B7 biotin.
No, yeah, exactly, biotin. Please do not forget these facts, guys. It is very important to remember. Okay, so now the location, the cytoplasm. You should know that glycolysis takes place in the cytoplasm. The HMP shunt, which is a very important pathway in the RBCs, takes place in the cytoplasm. The synthesis of cholesterol, you will see, and it will take place in the cytoplasm. You should know that smooth endoplasmic reticulum is involved in the formation of cholesterol. Okay. And the proteins, the ribosomes are in the cytoplasm. The rough endoplasmic reticulum is in the cytoplasm. Fatty acid formation takes place in the cytoplasm. Fatty acid oxidation takes place in the mitochondria, and formation takes place in the cytoplasm. The nucleotides formation, I told you, remember the bases, purines and pyrimidines, they all take place in the cytoplasm only. And some heme synthesis, some of the part of heme synthesis takes place in the mitochondria, especially the initial reactions, the formation of HeLa, amino levulinic acid, and the last part, the formation of heme, takes place in the mitochondria, and rest of the reactions are take place in the cytoplasm. The urea cycle, especially the formation of CPS, takes place in the in the mitochondria, and rest all reactions take place in the cytoplasm. The gluconeogenesis, initial parts, like I told you, this one, the conversion of pyruvate to oxaloacetate takes place in the mitochondria, and the remaining reactions will take place in the cytoplasm. Okay. I hope it's clear to you guys. Remember the enzyme transketolase, what vitamin it uses? Anyone remember it?
Thiamine. Thiamine.
Thiamine, right? Very good.
Thiamine.
It is a test of the thiamine, right? Test for the thiamine deficiency. Okay, so now we will move forward and study about these enzyme terminologies. There is nothing to explain, and I never encountered any question over these things. We just need to know the kinases will add the phosphate group, like I told you already. Remember these cyclin-dependent kinases? I told you that they will lead to the retinoblastoma phosphorylation, the addition of phosphate group. So all the kinases are involved in the addition of phosphate group, okay, and especially the ATP part. Similarly, the phosphorylase, they do not use the ATP but adds a phosphate. For example, the glycogen phosphorylase, it is a step takes place in the RBCs especially. You will study that later. The phosphatase, which removes the phosphate group. Okay, the dehydrogenase enzyme, which catalyzes the oxidation and reduction reactions. It can do both, either oxidize and either the reduces. The hydroxylase enzyme, which adds the hydroxyl group. Then the carboxylase enzyme, which transfers a CO2. And all of these carboxylase reactions require the biotin. The mutase, especially you need to know, the what was that acid is? Methylmalonic acid. Yeah, methylmalonic acid that converted into this, succinyl coenzyme A. It was a mutase enzyme that requires this vitamin B12. You should remember about it, okay? Then synthetase and synthase enzyme, they both join some molecules together to form a single molecule, okay? If you remember these terminologies, although not very much important, then you should know what are the rate limiting steps are in certain reactions, in some reactions. For example, in the glycolysis, okay, the phosphofructokinase 1, you will study that later in the later pages. That is a phosphofructokinase 1, which is a rate limiting enzyme. All these rate limiting enzymes are actually the slowest enzyme, all other enzymes are faster than them. And that's why you see that the rate of reaction is actually dependent upon these enzymes because they're very slow. Okay. So how much speed they convert the substrate into the product, it dependent upon these enzymes only, the rate of reaction. And that's why the rate limiting steps. Phosphofructokinase 1, which converts the fructose into fructose 6 phosphate into the fructose 1,6 biphosphate. This is the enzyme, the rate limiting enzyme. You should know that AMP activates it and ATP inhibits it, which makes sense because if you have lots of energy inside your body, you do not need a glycolysis, right? So ATP inhibits it. You will study that citrate also inhibit this enzyme because it, when you have too much of energy or too much of acetyl coenzyme, you need the formation of fatty acids inside your body. And citrate is going to inhibit this step, the glycolysis, and convert all of the substrate into the formation of fatty acids. So citrate is also an inhibitor of this enzyme. Fructose 2,6-biphosphate, you will study that later. Again, it is an activator of this enzyme. It forms by the phosphofructokinase 2. This product, this fructose 2,6-biphosphate is formed by the phosphofructokinase 2. And it is an activator of this enzyme. Then the gluconeogenesis, the fructose 1,6-biphosphatase step, which is opposite of this step. This is a rate limiting step in the gluconeogenesis. Of course, these both are the slowest enzymes. Then the TCA cycle, isocitrate dehydrogenase, you must remember, it is a rate limiting step. It gets activated by the ATP, which makes sense. You do not have energy, you require the TCA cycle. And it is inhibited by the ATP as well as the NADH, which makes again sense because if you have too much of energy, you do not need the TCA cycle. Then the glycogenesis, which is a formation of glycogen, it is dependent upon the glycogen synthetase. Okay, and you need to know that insulin actually promotes the formation of glycogen inside your body, which makes sense because if you have too much glucose, you want to store it. And if there is no glucose available, then glucagon will get activated. It will deplete these stores. Okay, and the glycogenolysis, which is the breakdown of the glycogen, again, it is activated by the glucagon and inhibited by the insulin. Okay, you do not have energy. That means you require the breakdown of glycogen. So AMP activates this pathway and ATP inhibits this pathway. Then the HAP shunt and this glucose 6-phosphate dehydrogenase enzyme, which is the same enzyme. If it becomes deficient, you won't get the NADPH and you will get this hemolytic anemia thing. You will get this glucose 6-phosphate dehydrogenase deficiency. Later study you will get. So HAP shunt, which is a rate limiting step, NADP will activate it and NADPH will inhibit it. Okay. Then the de novo pyrimidine synthesis, CPS2, you should remember, it's a rate limiting step in the pyrimidine synthesis pathway. Remember, in the second page of first aid, not the second page, third page of first aid, I told you about this enzyme, about the CPS1 location, the CPS2 location. So now, and then the de novo purine synthesis, this PRPP aminotransferase. What this enzyme does, remember guys? PRPP aminotransferase enzyme.
It catalyzes the formation of inosine monophosphate. Am I right?
Yeah, exactly. You're right. From PRPP to from PRPP to IMP. Okay.
IMP.
PRPP aminotransferase. You should remember the function. Okay. Then the urea cycle, CPS1 enzyme is a rate limiting enzyme. And the fatty acid synthesis, acetyl coenzyme carboxylase, you will study. It requires a vitamin B7 as a cofactor, remember? And again, if you have too much of glucose, you will have insulin. You want the glucose to be stored. The one way is to store in the form of glycogen, and the other way is to form the fatty acids. So insulin, and remember I told you about this citrate. When you have too much of acetyl coenzyme, your citrate will inhibit the glycolysis and will activate this, fatty acid synthesis pathway. The citrate is involved in these reactions.
You just said
Glucagon again.
acetyl-CoA carboxylase requires vitamin B7, right? Because it's a
Yeah,
carboxylase.
yeah, vitamin B7. Remember I told you?
Yes.
Or the carboxylation reactions require the vitamin B7. Remember? So all these carboxylase enzyme actually require the vitamin B7 to function. Then comes the fatty acid oxidation and carnitine acyl transferase, which actually transfers your fatty acids from cytoplasm to the mitochondria, is the rate limiting step. And you should know this malonyl coenzyme A, which is formed from this acetyl coenzyme A, by the action of this enzyme, it inhibits this because you need this step. Why? Because if you are forming the fatty acid, you don't want your cell to utilize it at the same time. So what happens if you are forming the fatty acids, if you are forming the malonyl coenzyme A, it will inhibit the oxidation of fatty acids. We will study that later again. The ketogenesis, which is the HMG-CoA synthetase, and in the cholesterol synthesis, HMG-CoA reductase. Remember these enzymes type. It is so